Dear students, Assalamu alaikum. Towards the later part of the last lecture, we studied reabsorption in the early distal tubule. The objectives of today's lecture are to learn about reabsorption in the late distal tubule and the medullary collecting duct. And secondly, the regulation of tubular reabsorption. So, continuing with reabsorption and secretion in different parts of the nephron, let's now consider important points about the late distal tubule and the cortical collecting tubule. Late distal tubule and the cortical collecting tubule have similar functional characteristics. They are composed of two distinct cell types. Number one, principal cells, and number two, intercalated cells. This figure shows cellular ultrastructure and transport characteristics of the late distal tubule and cortical collecting tubule. These parts of the nephron are composed of two distinct cell types, the principal cells and the intercalated cells. The principal cells reabsorb sodium from the lumen and secrete potassium ions into the lumen. Type A intercalated cells reabsorb potassium and bicarbonate ions from the lumen of the renal tubule and secrete hydrogen ions into the lumen. An important point to note is that the reabsorption of water from this segment is controlled by the concentration of antidiuretic hormone. Principal cells reabsorb sodium. This depends on activity of sodium-potassium ATPase pump in each cell's basolateral membrane. A low sodium concentration inside the cell favors sodium diffusion into the cell through special channels. Principal cells secrete potassium. This involves two steps. Number one, potassium enters the cell from renal interstitial fluid. Number two, potassium diffuses down its concentration gradient across the luminal membrane into the tubular fluid. Principal cells are primary sites of action of the potassium-sparing diuretics like spironolactone, eplerenone, amiloride, and triamterene. This figure shows the mechanism of sodium chloride reabsorption and potassium secretion in the principal cells of the late distal tubule and cortical collecting tubules. Sodium enters the cell through special channels and is transported out of the cell by the sodium-potassium pump. Some drugs compete with aldosterone for binding sites in the cell and therefore inhibit the effects of aldosterone, which are to stimulate sodium reabsorption and potassium secretion. In addition to these, sodium channel blocker drugs directly inhibit the entry of sodium by blocking the sodium channels. Now coming to the intercalated cells, they constitute 30 to 40% of the cells in the collecting tubules and collecting ducts. There are two types of intercalated cells, type A and type B. The intercalated cells play a major role in acid-base regulation, as we will learn in later lectures. Type A intercalated cells are important in secreting hydrogen ions while reabsorbing bicarbonate in acidosis. They secrete hydrogen ions by two types of transporters proteins. Number one, hydrogen ATPase transporter, and number two, hydrogen potassium ATPase transporter. Hydrogen is generated in these cells by the action of carbonic anhydrase on water and carbon dioxide to form carbonic acid, which then dissociates into hydrogen ions and bicarbonate ions. The hydrogen ions are then secreted into the tubular lumen, and for each hydrogen ion secreted, a bicarbonate ion becomes available for reabsorption across the basolateral membrane. As mentioned earlier, type A intercalated cells are especially important in eliminating hydrogen ions while reabsorbing bicarbonate ions in acidosis. The figure shows type A intercalated cells containing hydrogen ATPase and hydrogen potassium ATPase in the luminal membrane of the renal tubule. These transporters secrete hydrogen ions while reabsorbing potassium ions and bicarbonate ions in acidosis. The other type of intercalated cells, the type B intercalated cells, secrete bicarbonate ions into the tubular lumen while reabsorbing hydrogen ions in alkalosis. Therefore, they have functions opposite to those of type A intercalated cells. Type B intercalated cells have hydrogen and bicarbonate transporters on opposite sides of the cell membrane compared with the type A cells. So the hydrogen ions are actively transported out of the cell on the basolateral side. Remember, on the basolateral side of the cell membrane of these type B intercalated cells by the hydrogen ATPase. The figure shows the type B intercalated cells. In type B cells, the hydrogen ATPase and hydrogen potassium ATPase transporters are located in the basolateral membrane and they reabsorb hydrogen ions while secreting bicarbonate and potassium ions in alkalosis. Another point to consider related or regarding the late distal tubule and the cortical collecting tubule is their permeability to water and urea. With high levels of antidiuretic hormone, these tubular segments are permeable to water, but in the absence of ADH, they are virtually impermeable to water. This provides an important mechanism for controlling the degree of dilution or concentration of the urine. Regarding the permeability to urea, the tubular membranes of both segments are impermeable to urea. And this is similar to the diluting segment of the early distal tubule. Medullary collecting duct. Although medullary collecting ducts reabsorb less than 10% of the filtered water and sodium, remember they are the final site for processing the urine, and they play an extremely important role in determining the final urine output of water and solutes. The epithelial cells of the collecting ducts are cuboidal and have relatively few mitochondria. The special characteristics of this segment are: number one, the permeability of the medullary collecting duct to water is controlled by the level of antidiuretic hormone. With high levels of ADH, water is reabsorbed into the medullary interstitium, reducing the urine volume and concentrating solutes in the urine. Secondly, the medullary collecting duct is permeable to urea. There are special urea transporters that facilitate urea diffusion across the luminal and basolateral membranes of the epithelial cells lining the renal tubule. Some urea is reabsorbed into the medullary interstitium, helping to raise its osmolality, and this contributes to form concentrated urine. Thirdly, the medullary collecting duct is capable of secreting hydrogen ions against a large concentration gradient. This figure shows the cellular ultrastructure and transport characteristics of the medullary collecting duct. The medullary collecting ducts actively reabsorb sodium and secrete hydrogen ions and are permeable to urea, which is reabsorbed in these tubular segments. The reabsorption of water in medullary collecting ducts is controlled by the concentration of antidiuretic hormone. Let's now study the second topic of today's lecture, which is the regulation of tubular reabsorption. An important feature of tubular reabsorption is that reabsorption of some solutes can be regulated independently of others, especially through the hormonal control mechanisms. As it is essential to maintain a balance between tubular reabsorption and glomerular filtration, there are multiple mechanisms that regulate tubular reabsorption. These mechanisms include: number one, glomerulotubular balance; number two, physical forces of number one, peritubular capillaries; number two, renal interstitial fluid. Third mechanism is the hormonal control, and fourth is the sympathetic nervous system activation. Let's first study the glomerulotubular balance. Glomerulotubular balance is the intrinsic ability of the tubules to increase their reabsorption rate in response to increased tubular load. Glomerulotubular balance helps to prevent overloading of the distal tubular segments when glomerular filtration rate increases. And the mechanisms for glomerulotubular balance can occur independently of the hormones. Next, we study the role of physical forces in the regulation of tubular reabsorption. Now, how these affect or how they regulate can be understood by this flow chart. Hydrostatic and colloid osmotic forces affect the rate of reabsorption across the peritubular capillaries, as they affect the reabsorption across the glomerular capillaries and other capillaries in the body. These changes in peritubular capillary reabsorption in turn influence the hydrostatic and colloid osmotic pressures of the renal interstitium, which ultimately regulates the reabsorption of water and solutes from the renal tubules. So forces in the peritubular capillaries, they cause changes in the pressures of the renal interstitium, and these changes in the pressures in the renal interstitium, they regulate the reabsorption of water and solutes from the renal tubules. So let's study the peritubular capillary forces. The normal rate of peritubular capillary reabsorption is about 124 milliliters per minute. And reabsorption across the peritubular capillaries can be calculated as equal to Kf multiplied by net reabsorptive force. So what is this net reabsorptive force? The net reabsorptive force is the sum of hydrostatic and colloid osmotic forces that either favor or oppose reabsorption across the peritubular capillaries. The peritubular capillary physical forces include: number one, hydrostatic pressure inside peritubular capillaries, PC, which opposes reabsorption. Number two, hydrostatic pressure in the renal interstitium, PIF, outside the capillaries, which favors reabsorption into the peritubular capillaries. Number three, colloid osmotic pressure of peritubular capillary plasma proteins by C, which favors reabsorption. And number four, colloid osmotic pressure of proteins in the renal interstitium by IF, which opposes reabsorption. So you can see in this figure showing the summary of the hydrostatic and colloid osmotic forces that determine fluid reabsorption into the peritubular capillaries. It also shows estimated values for the humans, the net reabsorptive force is 10 millimeters of mercury, causing fluid and solutes to be reabsorbed into the peritubular capillaries as they are transported across the renal tubular cells. Regulation. There are two determinants of peritubular capillary reabsorption. Number one is the peritubular capillaries hydrostatic pressure, and number two, peritubular capillaries colloid osmotic pressure. So hydrostatic pressure and colloid osmotic pressure are the two determinants which regulate the reabsorption into the peritubular capillaries. The peritubular capillary hydrostatic pressure is influenced by number one, arterial pressure, and number two, resistances of the afferent and efferent arterioles. So let's study these factors one by one. First, the arterial pressure. Increases in arterial pressure, systemic arterial pressure, raise the peritubular capillary hydrostatic pressure and therefore decrease the reabsorption into the peritubular capillaries. Number two is the resistances of afferent and efferent arterioles, which influence the hydrostatic pressure. An increase in resistance of either the afferent or the efferent arterioles reduce the peritubular capillary hydrostatic pressure and therefore increases the reabsorption into the peritubular capillaries. The other determinant, the peritubular capillary colloid osmotic pressure itself is determined by two factors. Number one, systemic plasma colloid osmotic pressure. Increasing the plasma protein concentration of the systemic blood tends to raise the peritubular capillary colloid osmotic pressure and therefore increasing the reabsorption in the peritubular capillaries. The other factor which influences the peritubular capillary colloid osmotic pressure is the filtration fraction. The higher the filtration fraction, the greater the fraction of plasma filtered through the glomerulus, and more concentrated the proteins become in the plasma that remains behind. So, increasing the filtration fraction tends to increase the peritubular capillary reabsorption rate. Changes in the peritubular capillary coefficient Kf also influence the reabsorption rate, as Kf is a measure of permeability and surface area of the capillaries. Therefore, increases in Kf increase the reabsorption, and decreases in Kf decrease the reabsorption into the peritubular capillaries. With this, I end today's lecture. We will continue with the regulation of tubular reabsorption in the next lecture.